For our next section, we're going to start talking about the most complicated piece, which is adaptive immunity. Adaptive immunity, in addition to the divisions we've talked about before, we can divide into humoral immunity and cell-mediated immunity. So humoral immunity is the antibodies and the direct action of the antibodies. So antibodies go out, they bind to microbes, and that can cause problems directly. But then we have cell-mediated immunity, which is everything else, all of the stuff that the cells are doing to go out and fight these. So we're going to walk step by step through this whole process. So B cells are cells that sit around. They have very particular receptors, and when they find something that matches the receptors, it binds to them. But even if it's holding on to something that matches the receptor, it's not going to do anything right at first. So it's just going to sit there holding that antigen. And it's not going to do anything until it's activated by a T helper cell. So for it to do something, it has to both have an antigen and be activated by a T helper cell. Then the B cell will divide to produce plasma B cells and memory B cells. Plasma B cells produce antibodies. So those antibodies that bind to this particular antigen. So these antibodies start going out. We'll talk more about what exactly that is, but they're proteins that are going to bind to this antigen. Our phagocytes then prefer intruders that are marked with antibodies. They will eat other things, but they prefer things that are marked with antibodies, and they will go after those specifically. So once something's been marked with an antibody, then it's going to go target that. And the memory cells are going to hang around, and if that same thing comes back, then the memory cells will remember it and go, hey, wait, this is important, and start this chain much faster. Now we talked about helper T cells. What is a helper T cell? We said it activates the B cell. Well, great, so it activates the B cell. How do we get a helper T cell? How does it get activated? What makes it activate a B cell? So the way that this happens is first you've got a phagocyte that eats a bacteria. Remember I said that once it had digested it in that lysosome, there were little bits of it that are left. So those bits of that bacteria will go to the surface of the phagocyte and present on the MHC complex. So here we have a dendritic cell, made a bacteria, now it's presenting little bits of that bacteria. The helper T cell comes along and connects up with that and says, oh, this antigen is the issue. And then it will be activated. And that activated helper T cell can then go activate those B cells. So antigen presentation can happen in a few different ways. We can have direct presentation where an infected cell can present something. We can have cross presentation where an infected cell can then be eaten by a dendritic cell, or a dendritic cell can eat other things and then present those. Then we have what's cross-dressing, which is our professional presenter cells, which see something being presented and then pick it up and start presenting it to other cells. So any of these can result in this presentation that can activate these helper T cells. So this antigen capture, we've got these dendritic cells that are eating things, that are collecting things and being professional presenters. We've got macrophages that present, and we have these B cells that are memory cells and present and produce these antibodies. So this is our sort of basic function of the adaptive immune system and how this whole chain of presentation works.